This is online lecture 17 for Bio 240. Today we're going to finish discussing quantitative genetics, which we started last week in class. We're going to focus on broad sense heritability. So on Thursday we started talking about multifactorial traits that are influenced by many different genes, but also can be influenced by the environment. We talked about a method to estimate how many genes are involved in this trait using the frequency of individuals in the F2 that have a phenotype like that of the parent. And we also talked about the statistics used to analyze these traits, and we talked about normal distribution, the mean, the variance, and standard deviation. Today we're going to talk about measures of variance and how they're used to work out broad sense heritability, which is labeled as H2. And this gives us an estimate of the proportion of variance in a trait that's due to genetic factors versus the environment. So let's start by talking about what causes variance. And as we talked in class on Thursday, we know that there are genetic effects. For example, the number of genes that are involved in producing a particular phenotype. And we call that variance due to genetics, VG. We also know that there are environmental effects. We can call that variance due to environment, or VE. And finally, there are interactions between genes and environment. Which is variance GE. And an example of the interaction between genes and environment might be, let's say, blood pressure and diet. So a person might have a systolic blood pressure that, if they eat a healthy diet, is about 120, but if they eat an unhealthy diet, goes up to 150. They're genetically susceptible to dietary change, whereas another person might have a systolic blood pressure of 130, but when they eat an unhealthy diet, it only goes up to maybe 135. So they're genetically predisposed to having a higher systolic blood pressure, but it's less influenced by a dietary effect. So given these three types of influences, we can say that variance in phenotype, which is VP, is due to variance due to genetics, plus variance due to the environment, plus variance due to genetics and environmental interactions. So knowing this, how do we figure out how much of the variance seen for a given trait is due to genes and how much is due to the environment? We know what phenotypic variance is because this is what we observe. But the best way to figure out the variance of one of these terms is to eliminate the other terms. Suppose you had a population of individuals who were genetically identical, so they were all clones of each other, highly inbred. We could make the genetic variance 0. And this can be done with research subjects such as yeast cells or mice or certain other model organisms, Drosophila, for example. And in this case, the phenotypic variance would just be equal to the environmental variance. Since there is no variance in genotype, we don't have to consider the genetic environment interaction. Similarly, if we were able to grow our population in a completely identical homogeneous environment, then we could say the variance due to the environment would be 0. And in this case, the phenotypic variance would just be equal to the genetic variance. Again, we don't have to worry about the environment-genetic interactions because the environment is held constant. Of course, in reality, it's nearly impossible to hold environmental factors constant, even in a greenhouse or even in a laboratory where you have research mice. There are subtle differences in their environmental exposure that can have a big difference on phenotypic expression. So geneticists will also often use inbred populations to try to eliminate genetic variance and figure out the proportion of phenotypic variance that's due to environment. An example of this was Sewell Wright's experiments on guinea pigs, where he was looking at white spotting phenotype. So what Sewell Wright did is first he took just a general population of guinea pigs and he calculated the variance for this trait, for white spotting in the population. And remember that the variance formula we used last time is just a statistical measure of the sum of the difference between every individual and the mean squared over n minus 1. So, for example, he may have had a population of 100 guinea pigs that ranged in the number of white spots they had from 0 to 100, and there was a mean in there of something. So for every individual, he compared the number of spots that they had to the mean for the whole population, and then divided it by the number in that population minus 1. So when he did this whole calculation, he found that the phenotypic variance was 573. So he's actually able to quantify the variance in this population. Next, he inbred guinea pigs to get individuals that were almost 100% genetically identical. So they were homozygous for all of the genes that were involved in white spotting. And when he did this, he looked again at the population of inbred guinea pigs, and he found that the variance in the number of spots they had was 340. So in the first experiment, his phenotypic variance was genetic and environment, whereas in the second experiment, he could eliminate the genetic variance and just attribute it to the environment. Now, since Sewell's guinea pigs were growing up in the same laboratory environment, the first population, that environment should be the same as the inbred population's environmental influence, which is not 100% true, but it's relatively close enough for this type of analysis. So we're able to solve for the genetic influence as VP minus VE. And when he did this, he found that 573 minus 340 equals 233. So the variance due to genetics was calculated to be 233. Notice that for simplicity in this experiment, we dropped out the term variance due to gene-environment interactions. So this number here, 233 and 340, assume that environment and genetics have separate influences on the expression of white spotting in guinea pigs. But these numbers, 340 and 233, don't really mean a whole lot. A better expression is called H2, which is the broad sense heritability. And this is the proportion of phenotypic variance due to genetic influence. So we can write this as an equation: H2 equals the genetic variance over the phenotypic variance. So in this case, our genetic variance was 233. The total variance, the phenotypic variance, was 573, which is calculated to 0.41. And what that means is that 41% of the variance that was observed in white spotting was due to genetics. And then the rest of it, the 59%, is due to various environmental influences. So variance is Brodson's heritability is a very useful term in order to talk about genetics versus environmental influences. And the values for H2 range from 0 to 1, because it's talking about a proportion. So if H2 is equal to 0, it means that none of the variance in a population is due to genetics, right? Our top number would be 0, which means that all of that variance is due to the environment. And then conversely, if H2 is equal to 1, that means that all of the variance is due to genetics. So it wouldn't matter what the environment is like. If you inherit that gene, you're going to have that phenotypic expression. An example of this is hair color in humans. So it doesn't really matter what your birth experience is like. It doesn't matter what your early childhood development is like. Your hair color is determined by your genes. And if you inherit the gene for black hair, you're going to have black hair no matter what. And there's a lot more to the study of inheritance within quantitative genetics, but we're not going to have time to go into it in this class. A little bit about additive effects. We talked about additive effects last time, and you can actually quantitate the additive genetic variance. You can talk about dominant genetic variance, in other words, how epistasis might play a role, or how dominance or partial and codominance play a role in influencing variance. But we're not going to get into that here. It is important that you read about the limits and misconceptions of heritability in your book, in the sections on limits of heritability, and then I will see you all in class.